Hello dear students, welcome to this lecture on pentose phosphate pathway. Bernard Leonard Hocker is best known for his contributions to elucidating the pentose phosphate pathway. He was president of American Society for Biochemistry and Molecular Biology. He began his training in enzymology, searching for enzyme that would catalyze the reduction of cytochrome C by reduced NADP. This marked the beginning of his lifelong involvement with pentose phosphate pathway. Hocker's research interest turned to the enzymes that are involved in oxidation of 6-phosphogluconate, and he demonstrated that this pathway generates ribulose 5-phosphate. He played a key role in elucidation of pentose phosphate pathway, which included the discovery of enzymes like transketolase and transaldolase. His laboratory was the first to prepare ribulose bisphosphate and ribulose bisphosphate carboxylase, which are both essential components in photosynthetic fixation of carbon dioxide by green plants. In most animal tissues, the major catabolic fate of glucose 6-phosphate is glycolytic breakdown to pyruvate, much of which is then oxidized through citric acid cycle, ultimately leading to formation of ATP. Glucose 6-phosphate does have other catabolic fates as well, however, which lead to specialized products that are needed by the cell. Sharing the common metabolites, the oxidative pentose phosphate pathway, that's also called as hexose monophosphate shunt, can accomplish this task. The reactions are carried out by soluble enzymes that are present in cytosol and plastids. In this oxidative pathway, NADP is the electron acceptor, that's it accepts the electron from other substrates, yielding NADPH. Rapidly dividing cells such as bone marrow cells, skin and intestinal mucosa use the pentoses to make RNA, DNA and coenzymes like ATP, NADH, FADH2 and coenzyme A. In other tissues, the essential product of pentose phosphate pathway is not the pentoses but electron donor, that's NADPH, which is needed for reductive biosynthesis or to counter the damaging effects of oxygen radicals. Tissues that carry out extensive fatty acid synthesis like liver and adipose, or the cells that are very active in the synthesis of cholesterol and steroid hormones like liver, adrenal gland, gonads require NADPH that's provided by this pathway actually. Erythrocytes and cells of lens and cornea are directly exposed to oxygen and thus to damaging free radicals that are generated by oxygen. By maintaining a reducing atmosphere, that's high ratio of NADPH to NADP, they can prevent the oxidative damage to proteins, lipids and other sensitive molecules. In erythrocytes, the NADPH that's provided by pentose phosphate pathway is so important in preventing oxidative damage that a genetic defect in glucose 6-phosphate dehydrogenase, that's the first enzyme of the pathway, can have serious medical consequences. In the pentose phosphate pathway, we have got two phases. One is oxidative phase and second one is non-oxidative phase. First, we will deal with oxidative phase of the pathway. The first reaction of pentose phosphate pathway is the oxidation of glucose 6-phosphate by glucose 6-phosphate dehydrogenase to form 6-phosphoglucone delta lactone, that's actually an intramolecular ester. NADP is the electron acceptor, that's it accepts the electron and overall equilibrium lies far in the direction of NADPH formation. The lactone formed earlier is hydrolyzed to free acid, that is 6-phosphogluconate by specific lactonase. Then 6-phosphogluconate undergoes oxidation and decarboxylation by 6-phosphogluconate dehydrogenase to form ketopentose, that's ribulose 5-phosphate. This reaction generates second molecule of NADPH. Phosphopentose isomerase converts ribulose 5-phosphate to aldose isomer, that's ribose 5-phosphate. The net result is the production of NADPH, that's the reductant, and ribose 5-phosphate. The reactions of oxidative phase can be summarized as glucose 6-phosphate gets converted to 6-phosphoglucone delta lactone. The enzyme involved in this reaction is glucose 6-phosphate dehydrogenase. In this reaction, NADP gets converted to NADPH and H+. Next, 6-phosphoglucone delta lactone gets converted to 6-phosphogluconate. The enzyme involved in this reaction is lactonase. 6-phosphogluconate now gets converted to ribulose 5-phosphate. The enzyme involved in this reaction is 6-phosphogluconate dehydrogenase. Again NADP gets converted to NADPH. The enzyme glucose 6-phosphate dehydrogenase is the committed step in pentose phosphate pathway because 6-phosphoglucone delta lactone has no other fat except to be converted to 6-phosphogluconate. The enzyme lactonase catalyzes the hydrolysis of 6-phosphoglucone delta lactone. The product is 6-phosphogluconate. It is the irreversible step, but it's not rate limiting. 6-phosphogluconate dehydrogenase catalyzes the oxidative decarboxylation of 6-phosphogluconate to yield 5-carbon ketose, that's ribulose 5-phosphate. Here NADP serves as oxidant. Now the second phase of the pathway, that's non-oxidative phase. This phase recycles pentose phosphates to glucose 6-phosphate. In tissues that require primarily NADPH, the pentose phosphates produced in the oxidative phase of the pathway are recycled into glucose 6-phosphate. In this non-oxidative phase, ribulose 5-phosphate is first epimerized to xylulose 5-phosphate, that's epimerization takes place. Then, in a series of rearrangements of carbon skeleton, 6 5-carbon sugar phosphates are converted to 5 6-carbon sugar phosphates, completing the cycle and allowing the continued oxidation of glucose 6-phosphate with the production of NADPH. Continued recycling leads ultimately to the conversion of glucose 6-phosphate to 6 molecules of carbon dioxide. Two enzymes unique to pentose phosphate pathway act in these interconversions actually. First is transketolase and second is transaldolase. Transketolase catalyzes the transfer of 2 carbon fragment from ketose donor to aldose acceptor. Transketolase transfers carbon first and carbon second of xylulose 5-phosphate to ribose 5-phosphate, forming a 7-carbon product, that is pseudohaptulose 7-phosphate. The remaining 3-carbon fragment from xylulose is actually glyceraldehyde 3-phosphate. Next comes the transaldolase. It catalyzes the reaction similar to aldose reaction of glycolysis. A 3-carbon fragment is removed from pseudohaptulose 7-phosphate and condensed with glyceraldehyde 3-phosphate, forming fructose 6-phosphate and erythrose 4-phosphate. Now transketolase acts again, forming fructose 6-phosphate and glyceraldehyde 3-phosphate from erythrose 4-phosphate and and xylulose 5-phosphate. That's the erythrose 4-phosphate reacts with xylulose 5-phosphate and results in the formation of fructose 6-phosphate and glyceraldehyde 3-phosphate. Two molecules of glyceraldehyde 3-phosphate can be converted to fructose 1,6-bisphosphate, which ultimately gets converted to glucose 6-phosphate. The enzyme transketolase discussed earlier requires the cofactor thiamine pyrophosphate TPP, which stabilizes a 2-carbon carbanion in this reaction, just as it does in pyruvate decarboxylase reaction. Transaldolase uses a lysine side chain to form a ship base with carbonyl group of its substrate, a ketose, thereby stabilizing a carbanion that is central to this reaction mechanism. Now the reactions of non-oxidative phase of the pathway. Here first comes the enzyme epimerase. Interconverts stereoisomers, that's ribulose 5-phosphate and xylulose 5-phosphate. Now isomerase. Isomerase converts ribulose 5-phosphate to ribose 5-phosphate, that's used in nucleotide and nucleic acid biosynthesis. Both the reactions are reversible. Ribose 5-phosphate reacts with xylulose 5-phosphate to form pseudohaptulose 7-phosphate and glyceraldehyde 3-phosphate. The enzyme involved is transketolase. Now, glyceraldehyde 3-phosphate reacts with pseudohaptulose 7-phosphate to form erythrose 4-phosphate and fructose 6-phosphate. The enzyme involved here is transaldolase. Now, erythrose 4-phosphate formed earlier reacts with xylulose 5-phosphate to form fructose 6-phosphate and glyceraldehyde 3-phosphate. The enzyme involved is transketolase again. Finally, glyceraldehyde 3-phosphate and fructose 6-phosphate gets converted to glucose 6-phosphate through the enzymes of gluconeogenesis for entry to pentose phosphate pathway, maximizing the formation of NADPH. The reactions of non-oxidative part of the pathway are readily reversible and thus also provide means of converting hexose phosphates to pentose phosphates. The net result is the complete oxidation of glucose 6-phosphate molecule to carbon dioxide with concomitant synthesis of 12 molecules of NADPH. Studies of the release of carbon dioxide 14 from isotopically labeled glucose indicate that it is the glycolysis that is the more dominant breakdown pathway, accounting for about 80 to 95% of total carbon flux in most plant tissues. However, the pentose phosphate pathway does contribute to the flux, and developmental studies indicate that its contribution increases as plant cells develop from meristematic to more differentiated state. Now, the product of two oxidative steps is NADPH, and this NADPH is thought to drive the reductive steps that are associated with various biosynthetic reactions that occur in the cytosol. In non-green plastids such as amyloplasts and in the chloroplasts that are functioning in the dark, the pathway may also supply NADPH for other biosynthetic reactions like lipid biosynthesis, nitrogen assimilation, and others. Since plant mitochondria are able to oxidize cytosolic NADPH through NADPH dehydrogenase that's localized on external surface of inner membrane, some of the reducing powers generated by this pathway may contribute to cellular energy metabolism. That's electrons from NADPH may end up reducing oxygen and generating ATP. The pathway produces ribose 5-phosphate, that's a precursor of ribose and deoxyribose that's needed in synthesis of DNA and RNA. Another intermediate, the 4-carbon erythrose 4-phosphate, combines with phosphoenol pyruvic acid in the initial reaction that produces phenolic compounds including aromatic amino acids, precursors of lignin, flavonoids and phytoalexins. During early stages of greening, before the leaf tissue becomes fully autotrophic, the oxidative for pentose phosphate pathway is thought to be involved in generating Calvin cycle intermediates. Now, control of oxidative pentose phosphate pathway. The oxidative pentose phosphate pathway is controlled by the initial reaction of the pathway that's catalyzed by glucose 6-phosphate dehydrogenase. The activity of this enzyme, that's glucose 6-phosphate dehydrogenase, is markedly inhibited by high ratio of NADPH to NADP. In the light, however, little operation of oxidative pathway is likely to occur in the chloroplast because the end product of the pathway, fructose 6-phosphate and glyceraldehyde 3-phosphate, are being synthesized by Calvin cycle. Thus, mass action will drive non-oxidative interconversions of the pathway in the direction of pentose synthesis. Moreover, glucose 6-phosphate dehydrogenase will be inhibited during photosynthesis by high ratio of NADPH to NADP in chloroplasts as well as by reductive inactivation involving ferredoxin and thioredoxin. The synthesis of glucose 6-phosphate dehydrogenase is induced by increased levels of insulin to glycogen ratio after having a high carbohydrate meal. Insulin, when secreted in response to hyperglycemia, induces the synthesis of glucose 6-phosphate dehydrogenase and 6-phosphogluconate dehydrogenase, increasing the rate of glucose oxidation by pentose phosphate pathway. The synthesis of glucose 6-phosphate dehydrogenase is repressed during fasting. Glucose 6-phosphate dehydrogenase deficiency causes hemolytic anemia. Now, importance of pentose phosphate pathway. Pentose phosphate pathway produces NADPH and ribose 5-phosphate. Ribulose 5-phosphate may be converted to ribose 5-phosphate. That's actually a substrate for synthesis of nucleotides and nucleic acids and some other coenzymes. The pathway also produces NADPH. Glyceraldehyde 3-phosphate and fructose 6-phosphate may be converted to glucose 6-phosphate through enzymes of gluconeogenesis for re-entry to pentose phosphate pathway, maximizing the formation of NADPH, which is needed for reductive biosynthesis. 3-carbon glyceraldehyde 3-phosphate and 6-carbon fructose 6-phosphate formed by 5-carbon sugar phosphates may enter glycolysis for ATP synthesis. 5-carbon ribose 1-phosphate that's generated during catabolism of nucleosides also enters glycolysis in this way after first being converted to ribose 5-phosphate. Thus, pentose phosphate pathway serves as an entry into glycolysis for both 5-carbon and 6-carbon sugars. Importance of pentose phosphate pathway in erythrocytes. When erythrocytes are exposed to chemicals that generate high levels of superoxide radicals, reduced glutathione is required to reduce these damaging compounds. Glutathione peroxidase catalyzes the degradation of organic hydroperoxides by reduction, as 2 glutathione molecules are oxidized to disulfide GSSG. Pentose phosphate pathway is responsible for maintaining high levels of NADPH in erythrocytes for use as reductant in glutathione reductase reaction. Now, primary outcomes of the pathway. This pathway produces reducing equivalents, that's NADPH, which is employed in reduction reactions in anabolism, particularly in the synthesis of fatty acids. Generates ribose 5-phosphate that's used as a substrate for synthesis of nucleotides, nucleic acids and coenzymes. Erythrose 4-phosphate combines with phosphoenol pyruvic acid in initial reaction that produces aromatic amino acids, precursors of lignin, flavonoids and phytoalexins. In this lecture, we have studied about pentose phosphate pathway, its phases, its functions, primary outcomes of the pathway, how energy gets liberated during this pathway, and finally its importance related to biochemistry and some disease. So dear students, I hope that you have understood today's lecture well. Thank you.